Hi everyone, my name is Jacky, and I'm an associate professor at the School of Life Sciences of CUHK. Today I'm going to give you a brief introduction on some of the researches that are being carried out at our institute, and hopefully you will find them interesting and consider joining us. I'll specifically focus on a specialized field of research called structural biology. So what exactly is structural biology? In science, seeing is believing. Structural biology is a field of research that aims to obtain the three-dimensional structures of biological macromolecules, like DNA, RNA, and proteins. By seeing the structures of these macromolecules, we can try to understand how they carry out their functions. In particular, we are highly interested in protein structures because, as you may already know, they are the major workhorse that carry out most of the reactions in our cells. They usually interact with each other and work as a team when they carry out their functions. So it would be even better if we can see how different proteins form complex, because that way we can understand how they carry out the complex cellular functions that keep us alive and healthy. Let me quickly use some Lego to demonstrate what I mean. So this Lego truck here is built up from many different pieces of Legos, just like our proteins are made up of different amino acids. After seeing this Lego truck with our own eyes, we know the wheels are positioned at the bottom of the truck, so it can move back and forth. But what if the truck actually looks like this? This is built from the same pieces of Lego, but this actually cannot move. So now you may have a better idea on why we need to look at the structures of macromolecules. So let's take a step further. I have this movable truck and this gas station. Okay, so let's imagine these two are actually protein. So by looking at these two pieces, I know that the truck can park into the center of the gas station like this. It cannot enter from the other side. So same applies to the protein complexes that I talked about. Without looking at them directly, we cannot be 100% sure on how the two interact with each other. Now let's imagine these two, when they interact, when the truck docks into the gas station, disease can happen. So as structural biologists, what can we do? We actually can look at the structure, design a specific drug like this piece of Lego here, so that the drugs can specifically bind inside the gas station to block the entry of the truck. Voila, no more disease will happen. And this is why structural biology is so important. Sounds pretty straightforward, right? Unfortunately, it is not that easy to see the three-dimensional structures of macromolecules. This is because they are so small that we cannot see the details even if we use the best light microscope in the world. We need to rely on different scientific methods to visualize their structures. These methods include NMR spectroscopy, X-ray crystallography, and single particle cryoelectron microscopy. I'll talk about the cryo-EM a bit later. In case you still don't think that protein structure is important, let's take a look at the structure here. The two proteins you are seeing here, the green one on the top, and the one in cyan color at the bottom, are the reasons why you are listening to my Zoom presentation instead of meeting me at CUHK. What you are looking at is how the spike protein of SARS-CoV-2 binds to ACE2, a protein on the surface of our own cells. It's this interaction that you are seeing here allows SARS-CoV-2 to infect us. So think about it. If we can stop the interactions, we can stop the infection. Many drug companies right now are using this structure to design drugs and vaccines to fight against SARS-CoV-2. In fact, some of the vaccines that are working right now are actually designed based on this structure. So now I hope you are convinced on the importance of structural biology. Structures can help us to understand how our cells work and help us to engineer protein for biotechnological purposes and even design drug. This is why CUHK SLS has developed a strong research program in structural biology to tackle many important scientific questions. For examples, teams at SLS have been studying on how influenza A proteins affect flu infection and how proteins produced by the bacteria H. pylori help them to survive in our stomach and cause diseases like stomach ulcers. Using information obtained from different protein structures, we have also tried to genetically engineer crops like soybeans or rice to enhance their cultivation, or try to understand neurodevelopment. More excitingly, we have also developed new strategies to deliver drugs to cells and develop potential inhibitors for the treatment of different diseases like flu, cancers, and neurodegenerative diseases. So if you are interested in drug development, this is a research field that you should consider in the future. I've mentioned about the cryoelectron microscopy at the beginning. This is a breakthrough technology in our field, and if you have paid attention to Nobel Prizes, the Nobel Prize in Chemistry in 2017 was actually awarded to scientists who developed this technology. This technology allows us to have a great leap in our ability to determine the structures of large and complex macromolecules. We at CUHK have worked very hard to bring this technology to Hong Kong, and we are the first local university to install such instrument you're seeing here. We now have one cryo-EM instrument running and will soon install another state-of-the-art instrument called cryo-FIB, which can help to perform another sophisticated experiment called cryoelectron tomography. I'll talk about that in a moment. With the cryo-EM instrument in our institute, we are now capable of obtaining structures of very large and complex proteins like the ones I show here. This is an image of a protein complex that help the cells to recycle biomolecules. And this is a movie of a toxin released by the bacteria H. pylori that kills cells in our stomach. So with this information, we will have a better understanding of how biomolecules get recycled and maybe help us to design drugs to fight against H. pylori. Can we go one step further and use this technology to understand biology even better? The answer, of course, is yes. So, using the cryo-FIB I mentioned, which is a revolutionary instrument that can help us to prepare ultra-thin biological samples, we actually can perform 3D imaging using the cryo-EM machine on samples prepared by the cryo-FIB. This new technique is called cryoelectron tomography. These techniques allow us to visualize the 3D structures of the cells and organelles in their native states at very high resolution. So you can see that this is a very new and exciting era of structural biology. Hopefully, after you listen to my talk today, you will find this scientific field exciting and join us in the future. Thank you.